Ever popped an antibiotic and thought, who figured this out? Get ready to dive into one of medicine's biggest breakthroughs, penicillin. We're going old school, straight to Alexander Fleming's 1929 research paper. He lays it all out, this accidental discovery.
And let me tell you, scientific papers, they aren't exactly light reading, even for scientists sometimes.
No kidding. So picture this, 1928, Fleming's lab. He's studying staphylococcus bacteria, not exactly the friendliest little guys.
Yep.
But here's where it gets interesting. Fleming, he leaves out a Petri dish. You know, those little dishes scientists use to grow bacteria.
Right.
Seems even geniuses forget things sometimes.
Happens to the best of us, right? But back then, contamination, that was the worst.
Right.
Imagine no antibiotics to keep things sterile. Constant battle against the invisible.
And in this case, the invisible threw a curveball. Fleming comes back to his lab, and what does he find? This Petri dish, completely taken over by mold. But not just any mold. This one, it caught his eye. He even describes it in detail, starting as a white, fluffy mass, then turning dark green. It's like he's describing some new planet just growing right there in his lab.
That description, as simple as it sounds, it was key. That's how Fleming identified it. Probably Penicillium mold. And that is where things get really interesting.
So Fleming's staring at this moldy Petri dish, and he notices something strange, the bacteria. They're giving the mold a wide berth. There's this clear ring around it where the bacteria simply refuse to grow.
This zone of inhibition, as it's called, was a huge red flag, or should I say green flag, considering the mold. You see, back then, infectious diseases were a leading cause of death. A simple cut could turn deadly. Doctors had very few weapons against bacterial infections.
So stumbling upon something that could potentially stop these bacteria in their tracks, that's got to be a heart-stopping moment for a scientist.
Absolutely. Now, remember those meticulous descriptions Fleming made about the mold's appearance? That allowed him to identify it as likely belonging to the Penicillium genus. And that identification was crucial because it gave him a starting point, a clue.
It's like a scientific breadcrumb trail. But Fleming, being the meticulous scientist he was, didn't stop there. He started testing different types of bacteria, carefully noting which ones were affected by the mold and which ones seemed immune. In his paper, he even includes these detailed charts, tables 2 and 3, laying it all out. You can practically feel the excitement building as he's jotting down his observations.
And what's fascinating is that he discovered penicillin was most effective against a certain type of bacteria called gram-positive bacteria. It's just a way to classify bacteria based on their cell walls, by the way.
Yeah.
These gram-positive bacteria, they include things like staphylococcus and streptococcus, which can cause some nasty infections. But some other types of bacteria, like E. coli, which is gram-negative, were less affected.
So not a magic bullet that eliminates all bacteria, but still, finding something that targeted specific types of bacteria, that was a game changer, especially back then.
Absolutely. This was the beginning of targeted therapies. Imagine, instead of using harsh antiseptics that harmed healthy cells too, you could target the infection directly. That's what Fleming was starting to see.
This accidental discovery was shaping up to be something huge, but Fleming knew this mold held a powerful secret. He just had to unlock it, so naturally, he started experimenting like crazy, right?
Exactly. He heated it, filtered it, even played around with the pH levels, all trying to isolate the ingredient responsible for this antibacterial effect. He was like a detective dusting for fingerprints, only he was trying to capture the essence of this bacteria-busting mold.
It's like he's trying to crack a code to save lives. And he wasn't just content with what it was. He wanted to know, how does it actually work?
Exactly. And how fast it could kill bacteria. That was a big one. He started comparing it to the antiseptics they used back then, and let's be real, those were pretty rough.
Yeah, talk about overkill.
Right. And what Fleming found is incredible. Penicillin, it was different. It seemed to target the bacteria, but leave healthy cells mostly alone. That was huge.
So gentler on the body, and this is where Fleming's really thinking ahead. Even back then, he's suggesting penicillin, it could be used as a topical antiseptic. And remember, this is way before they're using it for infections inside the body.
Talk about ahead of his time.
Yeah.
And another key finding, which we can see in Table 2, Fleming realized penicillin, it wasn't a one-size-fits-all solution. Some bacteria, they were resistant to it.
Like they had some kind of force field up or something.
Exactly. But figuring out which bacteria were susceptible and which ones could resist, that was huge. It's like drawing a map of the enemy's defenses.
Love a good strategy.
Yep.
But hold on, there's this one detail that just makes you scratch your head. Fleming noticed that sometimes penicillin, it was actually more effective against certain bacteria when there were other completely different bacteria hanging around What's that about?
It's true. Seems counterintuitive, but it's this fascinating peek into how complex that microscopic world really is. And you know what? This weird synergy actually helped them isolate Haemophilus influenza. That's the bacteria behind the flu.
So this little detail that seemed so insignificant, it ends up unlocking even more medical breakthroughs down the line. Crazy how that works. So let's bring it all home. A messy lab, a forgotten petri dish, and a scientist with a keen eye. Boom! Alexander Fleming stumbles upon penicillin, an absolute game changer in medicine.
It really makes you think. What other breakthroughs are out there just waiting to be discovered? Maybe there's another forgotten petri dish in some lab somewhere. Someone just needs to take a closer look.
That's what I'm talking about. That's the beauty of science, isn't it? Always more to learn, always more to discover. Thanks for joining us for this deep dive into the world of penicillin. Until next time.